medical side, because we are just we are all normal people, just lay people for medical, but it's nice that learning from sir. And today also I'm going to give some just overview of this in medical imaging process and how we are going to deal with that as a computer science people, then how we can help the industry. That's what my talk would be today. Okay, can you start sharing the screen, ma'am?
Yes, sir.
Yeah.
Please.
Transfer the control. I think I need some.
Yeah.
If stop sharing, then I can.
Yeah, Bhavani, stop sharing. Jennifer.
Thank you.
Thank you.
Yes. Okay, so today's title I've given for just for our knowledge sharing is comprehensive cancer care through biomedical imaging. So I hope most of you are from this research domain and even from different research domains. I could see many doctorates are there and many research scholars are here. So it's like knowledge sharing, rather it's like preaching from one side or giving knowledge from one side. We can have discussions and we can go ahead. Okay, so today's agenda will be like this. Just cancer overview, comprehensive care, imaging modalities, biomedical imaging, nanotechnology in cancer care, nano-oncology, computer-aided diagnostics, top 4 technologies in medical imaging. Medical image, I mean, it's like a big data processing. So I have termed it as like a medical image, big data processing. So that's whatever. Then if time permits, then I can talk something about our work and about edge computing, especially during the process of any diagnosis, how the process, the diagnosis, the how edge computing can help for us. That's what I want. So you can have discussions. You can in between, as ma'am has informed earlier, we can have question session at the end after 5:15. In between also, if you want to interrupt, also no issues. It's okay, fine, no issues. Just if you want. So I want to have some discussion on this. How many of you know what is cancer? Just what normally I ask is like, if you know the answer, just put 1. If you don't know the answer, just put 0. Or in between 0.5, 0.7, up to your knowledge, you can grade yourself and you can put it just in the chat box. No need to talk, just you can put it in the chat box. Anyway, by the time I will go, just I'm seeing the, yes, beautiful. I'm getting first person is putting 1. Yes, doctor has given, Dr. Pankaj has given the answer. Was fine, sir knows that very well. Yes, many of you know. Okay, if I ask you to define, so what is cancer? It is a change of normal cells to the abnormal cells, and the abnormal cells start living, right, without control, and they divide and they multiply and they go. Actually, if you see in the real time, just for a research perspective, if you see, if I ask cancer why we are struggling a lot, the whole world is struggling, so billions of dollars are invested for this cancer diagnostics and cancer cure and cancer drugs. And you know that the current situation, if a normal person goes for cancer treatment, he may or she may need to spend around 10 to 15 lakhs for the chemo, as well as the medications and the beds. So there are so many things, continuous process, minimum of 6 months to 10, 12 months treatment. I'm talking about that. If God helps, really it's good. Otherwise, the things will change. So this cancer cells, why it is like that? It is our own body cells, right? It is not the virus. It is not like COVID coming from some other third party, like for the human beings, some animals or some other places. It's not coming, right? This is our own cells when they are the abnormal cells or just destroyed cells which are not killed properly, the malfunctioning cells. Then they start accumulating in our body, right? So it's like the spreading through the blood system and the lymph systems, and there are so many types of cancers. That's fine, but the major types will have more impact. and the other types, it's like, we can say the subtypes like that. So loss of normal growth control leads to cancer, right? The normal cell division goes like that. But you can see in the second part, cancer cell division, how it goes. You may ask why we have to know all these things, but because when we apply algorithms, what's happening, what to do, right? So all these things, we should know what's happening in the background. Just for information, just I'm giving. And one more thing, see the difference. Normal cell, how nicely it divides and multiplies, right? But you can see the cancer cell division here. When I see this in my mind, often I feel like that, நம்ம இதுல மூவில சொல்ற மாதிரி சிங்கம் வந்து சிங்கிளா வரும், பிக் தான் கூட்டமா வரும் இல்லைங்களா? அதே மாதிரிதான் இந்த இடத்துல, that's how this part, if you see, it is it's a crowded, right? It is multiplying fast. The negative negative thoughts, negative news will fast spread, right? But good news, it takes time. Good things, it takes time. So like that only you can imagine how it is in our own body becomes enemy to us, right? The cells. Okay, so main categories, I don't just skin related and bone related. This bone, not only the bone, cartilage, it's like when two joints are there, right? Bones joining that tissue, the tissue is there, and fat, muscle, blood vessels. Then leukemia is like bone marrow, you know that. Lymphoma, the cells of immune system, because lymph nodes, we say LYMPH, right? That lymph nodes and lymph vessels, which carry the fluid for which has the CBC, white blood cells, WBC, which is mainly for, we say, immunity, right? That fighting the negative agents, fighting the negative agents. Just a second. Fighting the negative agents, anything coming inside the flow, the vessel, right? So that the flow, the liquid fluid flows through the lymph vessels, and in the lymph area, it's stopping, and it is not permitting the cancer cells or inflated cells or any kind of viruses, right? These things are happening. So the immune system gets affected. The cancer that being the cells of the immune system, and central nervous system, brain and spinal cord. Just so you might have heard about it. Even yesterday, some doctor was talking about this spinal cord, right? The exactly that cancer started in that particular place, right? So in that particular area, it's not, does not it spread. He was giving one case study, if I remember exactly that part, right? So that central nervous system, so it depends on the area, it spreads across, and so causes and high risk, the cancer, then high risk populations consequences. I don't need to get into this very elaborately, but we see here they have given the general idea about this causes, right? Whatever be the causes, we can control it, but we are not sure that. So we can ask the question then. So if that is a person, a woman who is going through some kind of cancer, she never come across even a cigarette, even the smoke of the cigarette, but she has been diagnosed with cancer, right? So that is not in our hands. So this, some causes we say. Okay, so this thing, the causes and high risk population who are all, vulnerable to this cancer infection. I don't say infection; it is like cancer affection, affected by the cancer consequences. So as we know that it is really costly and it's very painful, and we know that how the people are, struggling in our own family. We might have seen somebody, right? So that's the staging. We say in a different, 0 to 1, we say. So summary staging, we can say in situation, in the situated, in particular that place, just it's we can say it's a beginning, local, regional, and distant. So based on this, we can we can say that the stage is like 0 or 1 or 2 or 3 and 4. So if it has crossed to different parts of the body, then you can say that it is stage 3 or 2, 3 to 4. That's a normal thing we say. All right. So these stages, how we can identify, that's a big challenge for us. That's a big challenge. So if normally doctor says if they had come in stage 1 or 2, then we could have saved the person, like that we say. And sometimes we say that the person that pancreatic cancer, if you heard about that, then it is a very rapid spreading one, and it is the lifespan is, even according to the data, the lifespan is very less nowadays. It's not like days are counted. So if you talk about some other cancers, normally cervical, sorry, ovarian cancers, like if they can be identified at some different stages only, maybe three, at least two, three, four. And in that stage only they were able to identify. It's really, it's like lucky. Then we can say it is found in the earlier stages, and they can be saved, and we can have very healthy. But it's all, that's one side. It is the reality. We talk about the reality, right? But the other side, it's we have to help, right? The one side, the physics stream is fighting to help with the devices. Other side, the biomedicine, biotechnology, and here we are also the computer science people. Also, we are there to try our best, to give our best through algorithms, to technologies, to analysis, analytics, and gaining the knowledge from different sources, like using artificial intelligence, right? So here you can see TNM station. We say that primary tumor, just extent of the primary tumor, absence or presence of regional lymph node involvement. That means lymph nodes, it's like we can say the near the below the arm, just at some places here, this place, and that lymph nodes where it is connecting, there also it is involved. Then absence or presence of distant metastasis. As I said, it's like once it is gone to the next place, right? We can say that it is next place is metastatic. It's if you want to remember exactly like what I can say normally when I tell my research scholars, like if you want to remember, like data is metadata. What is metadata? Is data about the data, right? So when you talk about that cancer, this cancer of the other cancer. So the origin is different. Yesterday, even um, one participant asked, like the kidney, both the kidneys are affected by the cancer, but the primary location, where is the primary location, where it started? So they couldn't tell, and doctor said it's 1 in 100, maybe 1%. Maybe it's difficult to say, but normally they can say that the location where it started is the place where we can identify. So, okay, fine. This in zero, stage zero, it's situation, and the stage one is early. The most advanced disease is the fourth stage. Of course, no, we might have heard about it. Okay, so here in the current year, that in 2022 itself, in US, the prediction estimated new cancer cases we see in males around 9,83,000, in females 9,34,000. Here, the most of the cases you can see that it's like 27% is prostate gland cancer. We can see here in this females, it's 31% of the breast cancers, lung, and here it's, you can see that what is the percentage, how it is decreasing, and in different parts, but see in new cases it's like 10 lakhs in US. And even country like India, yes, we are also leading in this count. So but we need, just before going to the next slide, just to take it in a very light way, I would like to say everyone wants to be healthy, right? You and I, everyone wants to be healthy. We pray for that, we work for that. Even see, we may earn thousands, lakhs, whatever it is. At the end of the time, when the health care comes, we don't mind spending everything. Right. So just I want to say the main just in if you say spiritually, the main kind we can avoid the cancer through some spiritual meditation activities, right? One thing, just one proven thing I want to tell, that some participants, the volunteers were tested for their immunity power from their saliva. Then around 50 people, they have been taken for this study, and they were sent to one room. There they were screening the movie of Mother Teresa, where they were helping the poor people, the bedridden, lepers, and downtrodden people. So 45 minutes they screened the movie. Then after that, when they came out of the room, just they tested their saliva again. Almost 70% increase in their immunity power. So this is what I was reading in, just somewhere in Google. So this, it's a proven that we can develop our own immunity. Even the same thing you could have come across during the second wave of pandemic last year, April, May. So everybody was telling more than that, you pray, do meditation, all these things, more than anything, everything that helps us more lot, right? Okay, everyone, my prayer, our prayer is like that. The human race should be out of these diseases, right? It's a very painful disease. So it's a very good topic you have chosen actually. Even ma'am's articles, all these things I have seen. That is really good actually. This cancer care is a very, very important thing, because we are helping someone, at least one person in the world somewhere, to reduce their pain through our knowledge. It's really awesome. Okay, so now let us move to the next part, is a comprehensive care. Comprehensive care, what I mean to say is like that, a complete thing. From the day one, the patient enters the hospital and till he goes with the healing, complete healing, I can say, completely healed, or in case if it's out of her, because we have not found the complete healing procedure. Normally, if you say, the doctors say it's a not curable, they don't use, I don't say not curable, they don't use the term curable, they use the term treatable. If you listen to them, they say it's a treatable. That means we can control it to some extent. So when it goes out of our control, our technology control, our medicinal control, drug control, then we have to go for palliative care. That means the person is nearing his end of her end of life, right? So we have to do the palliative. So even till then, we have some role, but we will see that. So level of care, it's like table, you assume here is the column and here is the row. Okay, so here you say the primary thing, secondary level, and tertiary. Okay, the starting part. So first level, what prevention you can do is the vaccination, right? If I remember exactly, even for cervical cancer, I could see some vaccines are there. So wonderful vaccines are there, HPV1 4, I think, like that. So that can be taken by children from 9 years, female children from 9 years up to 45 years, people can take that, so that they can prevent. Because as per the count, if you say the statistic, every 9 seconds in the world, one woman fall prey to the cancer of this cervical cancer. So we can avoid, right? We can at least, the vaccination is the first one. And next is the clinical assessment. When we go to the secondary thing, clinical laboratory, of course, the pathology, biopsy, then endoscopy, then medical imaging, where we have some role. Even the endoscopy we can have, even pathology also we can have. That is nothing like we can't have it, because we have to simplify the process of this testing. All right, medical imaging, then the nuclear medicine, where currently people are really trying to find some promising results. They want to get some promising results, like attacking targeted therapy. You might have heard about this targeted therapy of this particular location alone. There is a difference between chemotherapy and targeted therapy. Okay, just we'll see later. Then that happens in the time of diagnosis and screening. Then and then we have this surgery, when they plan for the level of based on this, when we come across this second level, then there is a thing for surgery. And even after surgery, there may be some few cells of cancer cells may be left off, then we have to go for radiotherapy. There are some cancers like ovarian cancers, the probability of recurrence is high, right? The cancer is high, so that time they have to they have to go for radiotherapy or systemic therapy. So likewise it goes. Then again follow up, and once the treatment is over, then We can see from the here again clinical assessment. After the treatment is over, the clinical assessment is done, and if there is any hope, that's okay, we can go ahead with the treatment, or we have to go for palliative care. The palliative care is nothing but the supportive therapy, just to reduce their stress, depression, their pain, their misunderstanding. That itself is a big area of thing, like how people will respond. There is a research area actually, the palliative care. Even we are having that, we contribute for that, computer science people, just we can contribute for palliative. Even the research is going on from our side also. Okay, so here then if it is needed, again clinical laboratory pathology nuclear medicine goes, then finally it's okay. The life care is going on, right? So where we lie from the diagnosis, the finding of vaccination, please keep in mind, especially the researchers, just I can tell, because other doctors, they definitely they would have come across all these things. Really, they will have very good knowledge of this. So this the research college, you can think of it from where you can position yourself and you can contribute something, because it's a huge area. It is not that small area. Even if you take one area of anything, just you can contribute. It's a great thing. Okay. Okay, then imagine modalities. Just I don't need to say tell much about that. Just initially, immediately the doctor will say, okay, we'll see the X-ray. Okay, just normally like bone fractures, arthritis, osteoporosis, infections, breast cancer, swallow. That's all this thing. Just like X-rays. I don't need to talk about that. It's general. It's part of this course. We can think. Okay, so then. A CT scan. Just when you talk about the CT scan last year, people said during the second wave there was a lot of confusion. The world was going through a lot of confusion during the second wave of pandemic. They said one. I have to say, even officially, the AIMS doctor said that one CT scan is equivalent to 1,000 X-rays. And my friend who is in Chennai, who is a radiologist, he is having his own clinic and own scan center. Because no, Peter, it is like 1:2 only. If you want, maximum the CT scan will take 2 to 3 X-ray radiations it will emit. So we don't need to worry about that. Okay, fine. Whatever, a series of X-rays to create cross sections of the inside of the body, including bones, bone vessels, and soft tissues. This is what happening with this. We know that injuries are from trauma, bone fractures, tumors, and cancers, vascular disease, heart disease, infection, all these things. When you talk about these injuries from trauma, not only the physical injury, the physical injury happens even from the not only the physical trauma, it's happening because of the mental trauma. Please keep in mind when you do the research, because clinically when they come, because the injury of physical trauma as well as and the mental trauma, both are different. So we have to see that case. Okay, so MRI is magnetic, right, resonance imaging, right? We know that. So magnetic fields we use and radio waves to create detailed images of organs and tissues in the body. And it takes almost 40-45 minutes or 30 minutes normally, maximum of one hour. Okay, so the problem is with people have different the fear, the CT, MRI, and that itself another thing. Now even people are working for that. We computer science people are helping into that. In artificial intelligence, there is one research area I can say that just how the and just cinematic print. Okay, I think I'll show the slide later. That is helping to find out the factors which impact the patient during the scan, even how they are not able to cope up. You might have seen, you may think that what is this? Even the people who have taken many MRIs or CT scans, they may think that nothing wrong. I mean, what is there? What is the difficulty in that? But the truth is, there are people, at least 5,000 of the people are struggling while going through this kind of modalities. Okay, MRI. Ultrasound, it's a simple thing, we know that. It is for gallbladder, breast lumps, and the genital prostatitis. Okay, whatever blood flow problems, monitoring pregnancy, and biopsies, all these. Okay, fine. So this is next part is PET. Yesterday, sir was talking, doctor was talking about that very beautifully. That positron emission tomography. It is it's currently in current scenario of medical field, they are using it effectively. This part to detect the cancer. Not only the cancer, we can say the other issue, the problems like you can see, diagnose cancer, heart disease, coronary artery disease, Alzheimer's diseases, epilepsy, Parkinson disease. See, this is very beautiful, but it will work with the radioactive drug. Okay, though they will normally they inject through IV, intravene. They inject the radioactive material to our body, and it works with our tissues. It works with the depends on the thing, right? If you take if you take cancer, they say the fluorodeoxy glucose, that material is FDG sent to the body, and the characteristic of cancerous cell is like it consumes more glucose. Okay, it consumes more. That cell consumes more glucose, so that we can identify from there. Okay, so to swallow or have a radio tracer injected into a PT scanner room, which reads the radiation gives up by the radio tracer. There are two things. Just we'll see. Anyway, I think maybe next slide I will have. Imaging modality is like anatomy, physiology, molecular level. Just anatomy level and physiology level. CT you can say, ultrasound. MRI goes up to the molecular, and PET goes to the molecular, and optical also molecular. So when we talk about this molecular, it has more advantages currently. You can see that. So I don't need to talk about the top parts. Okay, this is like for our reference I have taken. I can give it to ma'am. You can collect this PPT anytime. So here I talked about this. We have copied this PET. All right, we have high sensitivity, strong penetration depth. The disadvantage is poor spatial resolution, radiation risk is there. Normally, currently people say that the radioactive element is injected in the body will have just 1 or 2 hours. What actually they say is at least 12 hours. The person who goes through PET scan, has to be away from, especially the pregnant ladies and the feeding mothers, the sick people, because the radiation emitted from their body also may affect. So at least 12 hours they say they have to be away from there. Okay, even aspect is the molecular high sensitivity. Then there also same kind of issue. Just we'll see later. Ultrasound, all these things are there. Here you can see the combination of this. That's very effective. I hope the doctor discussed yesterday. So this combination of the modalities, this will have more effective than the single one. We go with the single. Okay, please. So nuclear medicine imaging. So what we do is just a radiopharmaceutical is administered. Okay, just it's given to the person, and it is localized, and the tissue absorbs, right? Absorbs, and it is localized. That based on the tissue's characteristic, it absorbs and responds. From there, we get the gamma ray photons, and there's a radioactive decay of the radiopharmaceutical. Gadolinium, if I'm not wrong, actually I remember the one of the contrast agent. Right? Gadolinium is given with MRI, I guess. And here in PET, I said one is FDG, and fluorine 18 is given with the PET, and aspect is with another thing. Just I forgot. I'll remember that. I think it's nitrogen 99, I guess, if I'm not wrong. Nuclear medicine imaging is used to map physiological function, right? Perfusion and ventilation of the lungs, blood supply, so all this. So this, since we are injecting the IV, the radioactive material in the IV, it goes through the whole body. Right? Wherever the tissues are, the cell, suppose for example, particular cancer, they say, right, that cancer cells, wherever it is spread. Right? So that there you can see the finally the block shaded, the black color shaded one, you can get it. The one is the cancer cell where we have to treat. Okay, fine. The aspect is detect single photon emission by radioactive decay. Just I'll give one small video also later after this. So it goes to 64 to 128 projection spanning. Okay, so there, if you take the PET rate is 2, here it is 64 to 80, 128. Around the vision, individual scan lines from the projected images. Okay, fine. So this is positron emission tomography, and isotopes of carbon, nitrogen, oxygen, fluorine. Yeah, right? I mean, positrons are suitable for nuclear medicine image. Okay. See here, the what is based on the. So I am telling you this all. Why I am telling this? Because Saturday I think you are going to have one from Siemens. Okay, they are also developing just mainly the machine with artificial intelligence. I guess even with the augmented reality, they are going to they are developing for some particular cancer. I guess if I'm not wrong, so that is no, it will, we have to understand this. Given this one, you can take it as a research area, and you can work on two annihilation photons produced at 511 kilowatt, and emitted in opposite direction when positron loses its kinetic energy and combines with an electron. So it's just okay. So just I will play this video, just you can see just 1 or 2 minutes maybe.
Meet Dave. He's about to have a brain scan. Using the latest PET imaging technology, doctors will see Dave's brain at work. It's all possible because of tiny tracers, specially designed radioactive molecules. The PET scanner tracks the position of the tracers inside Dave, revealing his brain function in 3 dimensions. The tracers don't come from a stockroom shelf. Each one has to be made fresh at a nearby laboratory. This is Dr. Miller. He's making a tracer for Dave's PET scan. First, he needs to produce the radioactivity using a device called a cyclotron. It smashes high-speed particles into ordinary atoms, transforming them into new radioactive atoms. Now the clock is ticking. The new atoms are giving off their radioactivity, becoming plain old regular atoms again. There goes one. It means Dr. Miller and his team have to work quickly to build the tracer, a biological molecule. This is what makes PET so unique. The molecule could be anything: a sugar, a protein, a hormone. Each one is tailored to a specific function in the body and will show doctors where in the body that function is happening. Dr. Miller's team runs special chemical reactions to create a tracer from a drug used in the brain. The new tracer must pass quality control. It is purified and checked that it will function properly. The tracer is ready to use. Good work, Dr. Miller. The precious custom-made molecule is rushed to the PET clinic. Just the right level of radioactivity is measured out, and it's injected into Dave's bloodstream. There'll be a sharp scratch. It's not long before Dave's brain has absorbed the tracer, and the clever chemistry gets to work. Inside Dave, an incredible reaction is taking place. The radioactive atom on the tracer loses its radioactivity, giving off a subatomic particle called a positron. It's just like the tiny negatively charged electrons found on all atoms, except that the positron is positively charged. So when the positron hits an electron, at just the right speed, they combine and destroy each other. The energy is released as 2 gamma rays that travel straight out of Dave's body in opposite directions. The PET scanner is like a big ring of Geiger counters. When it detects 2 gamma rays on opposite sides of the ring, the doctors can calculate where in Dave's body the tracer must be. By detecting thousands of these events every second, and with some clever computer processing, Dave's brain activity is revealed in 3 dimensions, something no other imaging technique can do. For Dave, it was just a small injection. It wasn't so bad, huh? All made possible by the incredible technology of the tracer. The potential for PET is enormous. Different tracers can detect cancer, test heart function, and even track Alzheimer's. The PET scan has revealed Dave's brain is functioning as it should. Dave, you're A-OK. In the next hour or so, his radioactivity levels will be back to normal. It's another successful job for the incredible tracers. But the work of Dr. Miller goes on as he continues creating new tracers to help those in need.
Okay, so in this video, I would like to give you another research idea. Okay, so the two gamma rays from two gamma rays is spreading from that position, right? So here itself is like you can work on one research area, how it is identified and that accuracy of that storage in depicting as image. Okay, that is one area. Even the companies are working on that. That's why new machines are coming out, right? So it's not just like one machine is perfect, or one company brand, one brand that scanner is good, or the other one is not good. It's not like that. We are improving, right? Day by day, we are improving. Whether we can improve with that, because this is inclusive of like physics, then molecular biology, then of course our computer, because our computer science, the algorithms, without that we can't do anything like. So algorithms, it's in our hands only. We can have multiple algorithms, or we can have one single algorithm or combination of algorithms to give the better. See, biomedical imaging is okay, fine. So that's a comprehensive cancer care. It's a main pillar, right? It has many advantages. Right? It was over a wide range of time and size scales involved in biological and pathological processes. Time scales go from milliseconds for protein binding and chemical reactions to years for diseases like cancer. Size scales go from molecular to cellular to organ to whole organism. Right? So it's a study like the current. I'm sorry. The current role of imaging cancer management, you can see that developing molecular structure, signature, I'm sorry. There it is for screening, then diagnosis and staging, treatment and monitoring, then follow up, then again coming back, after treatment, is there any progress? Again, we are good to go. We have to go for diagnosis. If you take the PET scan, this is what happening. Once the doctor doubts that cancer is there with the, because of there are marker tests. You might have heard about different marker tests. One example I can say is like CA125. They say it's a blood test. It's a blood test. The CA125. That is like you know. That's for ovarian cancers. Okay, to find the ovarian cancer in the blood, so the value should be below 30. Sorry, no, it's below 50. 30 is a beautiful, wonderful value. If it's even below 50, is considered as normal, and above 50, then it shows that there is a high probability of ovarian cancer in that particular patient. Okay, fine. So what we do is like the PET scan is taken after the doctor doubts clinical diagnosis, the doctor doubts, then we are sending the patient for PET scan. Then they take the scan, they give the treatment, then again send for the PET scan to find out. Yesterday, if you remember exactly that one, the doctor was showing that one patient initially, how that throughout the below, the abdomen and the chest, even the lower abdomen, how the cancer cells had spread across. After 9 months of treatment, how it came down, and 4 years, after 4 years, how it had come down drastically like that. Okay, so this is a flow here now in this. Second, that future role, what we have to is like genetic predisposition, that screening, then DNA mutation, even just it started actually, it's happening currently. DNA mutation, pre-symptomatic therapy, then disease regression we have to find, then follow up again, going back to this. This is using this molecular imaging and quantitative. Normally we do with qualitative, right? So this is what we do, right? But it's inclusive of quantitative, right? That's what we do. Okay, fine. So integrated cancer care is with the detection, diagnosis, treatment, everything, again and again, sensing, data analysis, effector action, then communication, judgment, all this. It's actually it's like a package. The whole package is coming with this. So like every area people have to work, otherwise it will not be successful. This is such a very challenging part for even not only the patient, it's for the medical professionals, even the caregivers, and the people of like us. Uh, So if you just okay, before I'll tell you this, biomedical imaging all phases of cancer management, like prediction, screening, biopsy guidance for detection, staging, prognosis, okay, and therapy planning, therapy guidance, therapy response, recurrence, palliation, all these stages. Prediction is from the day one when the patient comes with the complaints of some kind of illness, and screening and to find whether to confirm, then biopsy guidance for detection to confirm to some extent, just taking the tissue of that particular region, then take just going for the checking, the testing, then finding the stage 0 to 4, then prognosis of this disease, then therapy planning, therapy guidance, therapy response, recurrence, palliation. Okay, everywhere we have our role. Fine, nanotechnology in cancer care is the next part. Just I'll give one small video, then we will go ahead.
Imagine something 80,000 times smaller than the breadth of a ridge on a fingertip, unlocking a new frontier into cancer research. Nanotechnology, the science of building small, is changing the way we look at cancer. More importantly, the way we look at diagnosis and treatment. Nanotechnology allows researchers to build new tools that are actually smaller than cells, giving them the opportunity to attack cancer at the cellular and genetic level. This technology not only enables health practitioners to detect cancer earlier, but also holds the promise of stopping cancer before it even develops. This revolutionary approach is so precise, doctors will be able to design a unique treatment for an individual's own medical and genetic profile. Based on computer chip technology, diagnostic devices such as nanoarrays are thousands of times more sensitive and accurate than current techniques. Because of their size, multiple lab tests can be done more rapidly and at a much lower cost, using one nano device instead of many. Nanoshells can be linked to antibodies that recognize tumor cells. Once they are taken up by the cancer cells, near-infrared light is applied, killing only the tumor and leaving neighboring healthy cells intact. Scientists are engineering nanoparticles such as dendrimers to seek out and destroy cancer cells. This amazing technology can be customized for targeted drug delivery, improved imaging, and near real-time confirmation of cancer cell death. Moving research from bench to bedside.
Okay, fine. Just I want to show. This is about their institute they are showing. Okay, fine. See, if you talk about this nanotechnology, if nanoparticle, if you say the one, so if you take 1 inch of size, there if you divide it by 2,54,000, right? 2,54,000 partitions if you make in 1 inch size, then that one particle is the nanoparticle, right? So that one particle size, right, we are working on that. And the other side, if you see the infrared, what they are sending only on this particular cancer cells, because this nanoparticle will observe our I'm sorry, nanoparticle will be sent to the body, and this is absorbed by the cancer cells, and the infrared rays are attracted only by those particles, so that the particles which are residing inside the cancerous cells, so it's attractive, and cancer cells nucleus is burst. So it's like the cell is totally destroyed, the cancer cell is totally destroyed. It is like targeted therapy, we can say. Because normally in current thing, if you talk about chemotherapy just they inject the medicine, right? When they inject the chemo medicine, right, it's when it is injected, it travels through the whole body top to bottom. Even you might have seen the cancer patients immediately within 3 cycles of medicine, 3 weeks of medicine, normally they are dividing the cycles into weeks. Normally they give 3 cycles, then surgery, then 3 cycles. That's a trend, right? Or sometimes it depends on the patient's healthcare. They say they will complete all the 6 cycles, then they may go for surgery. And each cycle is divided into 3 weeks. 3 weeks dosage, 3 dosages, we can say that. So after the 1 cycle or 3 weeks of medicine, they'll start losing the hair. Almost they lose all the hair, right? So they are, even that is no cooling therapy is trying now there, but it is not very promising. It's not giving promising results. So they have to, what they can do is like, they have to go with that, the alternatives, like they have to accept that there are so many things and side effects. You can see the toxicity. Definitely they may have the vomiting, nausea, and they will have the diarrhea. They will have the weakness, and so much of energy loss, and you can see the tremendous loss in their healthy cells. So what we want to do in this nano oncology, we can say we are going to see nano medicine is applying just for only the targeting the cancerous cells, so that the cancer cell is destroyed. Normal cells are not touched. That's what. But that is we haven't succeeded. I don't say that we have completely succeeded in that. That part is still, we are working on that. We are helping that, biomedical people have to help, and physics people have to help, and chemistry people have to help. Yes, of course, the chemistry people, I forgot to tell. Then the computer science people, we are the, very big, because because of us only they are able to measure it accurately. If we are not there, they can't do anything. Okay, so just we can probably say that computer science people are really, we are giving the best to the world actually. So the treatment, diagnosis, detection, signaling, and treatment with the nanomedicine. Okay, fine. So focused research, clinical relevance, and team science. Gold nanoparticles is like the size, the nanoparticle, the silica. The silica is coated with the gold plated, you can say, the gold coated nanoparticle, or the silica of almost 20 nanometers. Just you can imagine nanometers, just what I said. The 20 nanometer size of that one is sent to the body. It's same thing like how I said, it is going to that particular tissue, it's getting dissolved in that, and working on that. And one more thing, see, suppose the the patient is diagnosed with cancer, and that is inner part of some tissue. Then what, the invasive surgery, right? We have to do that. We have to do whatever be the modality they use, that the surgery is just like they have to cut it, and to reach the inner part of that, they have to cut the outer part, and they have to reach the inner part. But this nano medicine, nanoparticle we are using for the nanomedicine, it goes directly to that cell and it kills the cancer cell. It does, we don't need to cut and cut the other tissue parts, or just it's like that. Okay, fine. So cancer nanotechnology, it involves, that's what I want to tell, biology, chemistry, engineering, physics, medicine all together. So it's quantum dots. Okay, the semiconductor quantum dots are there. Iron oxide nanocrystals, carbon nanotubes, polymetric nanoparticles, whatever it is they can use. So here you can see imaging, therapeutics, and diagnostics, detection, and informatics. So all together, with the nanotechnology depends on all this, that informatics will come together, and we can work together. We can attack on this cancer disease, and we can help. Unique structural, optic, and magnetic. So targeting and imaging, emission wavelengths, just like how we are, high molar extinction coefficients, conjugations with copolymer improves biocompatibility, selectivity, and decrease cellular toxicity. So that's the main thing with molecular cancer imaging. Just early detections we can have really in this, because in the beginning itself. Now one interesting thing is, I haven't put it in my slide, but I would like to say, they suppose a patient person is identified with some particular cancer, they can, the patient can go with the DNA test, so that if that value is above particular something, just I'm not very clear about it, just you can Google it and find out. Then they will ask their children, if they are having children, and then there is a high risk for the children to get the same kind of cancer, whatever it is. So that is like genetic. No, normally we say in one of the factors for cancer is like genetic one. So this blood test, they have to go for that. Then they can find out just even the children whether they will have the chance of getting this kind of cancer disease. Okay, nanowires, it's like the same thing like metallic semiconductor, polymer composite nanowires, functionalized by legends such as antibodies and oligonucleotides. Okay, capturing the targeted molecules, the nanowires changes the conductivity but up to 10 into 10 power minus concentrations. That's a beautiful thing. So we can just people are working on that, right? Just for our end. So how we can contribute? It's like only by gaining their knowledge, our algorithms will work. So active targeting, what I talked about is targeted therapy, right? So nanopractical particle how it is working. Even this in US, they have approved the Food and Drug Administration approved Abraxane and album that is albumin and paclitaxel. If you see the chemo medicines, one of this is like paclitaxel, right? You can see that. So albumin paclitaxel nanoparticle drug for the breast cancer treatment. Okay, it has been approved and it is working. Taxol and so you can see the cancer drug. Yes, like this is the same thing. It's a common thing. No, this is what they are working on. Okay. The future direction, the first one is development of nanoparticles as monofunctional, dual functional, trifunctional, multiple functional probes. It is like, you know just how it works with the disease diagnosis and if it is diagnosed, because when you take the PET scan itself, just we can imagine the situation like this. When the doctor diagnoses that the radiologist finds that the patient is with some cancer at the time of PET, he himself can inject another medicine, then nanomedicine, and he can start the process with the consultation with the chemotherapist. Now currently we say that chemotherapy doctors, chemotherapists, they are exclusively for that. Or the surgeons may come into the picture. If we really succeed in this, then surgeons may not be required in the later stage. Surgery is not required, so surgeons are not required. So the second direction is to study nanoparticle distribution metabolism, execution, and dynamics in vivo animal models. These investigations will be very important. We'll see that because I want to show the other parts. Just I'll go a little fast. Okay, so nano oncology is one exclusive thing. Many potential applications of nanotechnology in medicine. So cancer diagnosis and therapy remains the most significant, has led to the development of new discipline of nano-oncology because we focus exclusively. Nanotechnology can be used for different purposes, but here we want to use it for the treatment, diagnosis, treatment of oncology related thing, diseases, right? Cancerous diseases. So this is what the nanoparticle how it looks. Just you can I told just like you can remember like gold plated silica, right? It's a polymer. This cancer medicine is there, that's a nanoparticle injected that is going to work, penetrate the cancer cell and is going to destroy that. So chemotherapy, cytostatic drugs damage both malignant, that's what we know that. No, but we want targeted therapy, targets the malignant tumor alone is very much needed currently. Okay, so this compared with conventional drug delivery, nanoparticle mediated delivery of anti-cancer drugs. Such a wonderful research is going on in this part, is to bring out the best treatment for the patients, to reduce their pains, and to get the best result to cure the cancer wherever it is. So drugs delivered by nanoparticles may have a longer biological life due to the packaging protection and concentrated in the site of cancer. So here, that is the one good thing. There are things we have to understand. It is like we algorithms. Normally, we say time and the space trade-off, right? Normally, when we write the small algorithm, simple algorithm, we say time and space trade-off. But the thing is, here also, when longer biological life, that we have to take care how much that research is very much. Whether we this survival in the bloodstream, how many months depends on the patient, because it is a very challenging part how long they can keep it and how it will be affected. But the problem is we have our advantage of artificial intelligence, right? We have to use that, right? How we can do that, we'll see later. Nano oncology. So EPR is caused by the leakiness of tumor vasculature as well as poor lymphatic drainage. Just I should just therefore nanotechnology increases treatment efficacy, decreases side effects. Okay, so the past various diagnostics and drug delivery systems have been developed for the cancer therapy. So it is diagnosis and prognosis to improve the therapeutic efficiency. So theranostic strategy. Just I'm there are a few slides I had put, but I don't want to say everything. It is the combination of all modalities of this, and we can say dual functions. No, we talked about dual functions or multiple functions into one nanomedicinal system. We want to bring it together. Simultaneous drug therapy and monitoring of pathological process, therapeutic efficacy with medical imaging tools. So we want to integrate. Just this I wanted to say because we talked about the comprehensive cancer care, right? So we have to bring it together, multiple functions into one nanomedicinal system, right? That's what we want to have. So it's diagnostic and therapeutic designs, timely tailoring, okay, that we can give exclusive for that particular patient. See, now if you say chemotherapy, it is only for the, it's a common thing. Dosage may will be given the same kind of dosage and all these things given for the patients of that same kind of diseases. But it is we want only the particular patient specific. Right, here if you talk about Dolo itself, you can say I can take Dolo 650 and you can take even a child of 50 kg, 40 kg or 50 kg, he can take Dolo 50 because they depends on the weight we are giving that medicine, right? And 4 to 6 hours gap we are giving that. So it's like, physical properties. There has been considerable interest in the development of variety of functional inorganic nanoparticles in biomedical technology. Okay, so which have the advantages of visualized by MRI, where it coincides by an external magnetic field, or we can, because of the magnetic field, we can move that one, even that we identify that. Just that's what we combine everything, no. So this also works. The molecular is also working, magnetic part is also working. The one side just provide hypothermia, then in order to fully exploit their potential, magnetic particles are often engineered by conjugation. Okay, fine. Just I want to just small video, just 2 3 minutes maximum.
Scientists are using cells isolated from human tumors to develop new ways to tackle cancer. Here we can see the cancer cells growing in the culture dish in the laboratory. For most drugs to be active, they must go inside the cancer cell. Scientists are developing tiny particles, nanoparticles, that can be loaded with drugs. The nanoparticles can be modified so that they attach to the cancer cell and get carried inside. To make sure that the drug-loaded nanoparticles only attack the disease cells, they are engineered so that they only bind to the target cancer cells and not to neighboring healthy cells. Once they bind to the cancer cell, they are taken into the cell. They can't pass through the surface. Instead, part of the cell surface forms a pit and then it engulfs the nanoparticle. Now we are looking inside the cell. Cellular proteins wrap around the vesicle and drive it inside the cell. You can now see a vesicle carrying the nanoparticle moving towards the very center of the cell. The vesicle carrying the nanoparticle travels further inside the cell and then fuses with a compartment called an endosome. This is part of the digestive system of the cell's stomach. The inside is acidic, and its job is to digest incoming material. The nanoparticles are degraded inside the endosome, releasing the drug into the cell. The drug can then kill the cancer cell. Nanoparticle-based drug delivery provides a way of attacking cancer without the side effects of conventional chemotherapy.
Okay, so just about the nano drugs I wanted to say. Now just computer aided diagnostics, we know that. What is medical imaging? I'm very sure that all of us know that in this part. Just I'll go rush through. Okay, so this imaging chain, it's like we are getting the raw data and filtering the raw data, reconstruction of the image, then process the image, then analysis we are doing. So this part we know that, right? Because those who are in image processing, or those who have started their research in image, that definitely we would have studied this process. Normally we do this segmentation, right? So first we do the pre-processing. We remove the noises from there. Then we convert as sometimes digital, the image to digital form. Then again we work on that. Even you might have used the simple tool of MATLAB, right? We might have used it, but we have to go beyond all these things. Even though the beginning level we have to have all these things, the turning an image into data, okay, fine. Then we do the feature extraction, right? What is the ground truth image, what is the normal image, then how we can compare this ground truth and ground truth image of the normal, and the infected image, or the image with some abnormalities, right? So the qualitative features, quantitative features, and semi-automated, automated. So we do the examination of these features, right? It's like all these things we know: image segmentation, classification, and diagnostic characterization, content-based image retrieval, image annotation, right? And so it's made by a radiologist when the output of computerized image analysis method has been incorporated into their medical decision making process. There are softwares, there are many softwares provided by different companies to the hospitals, radiologists. They are using just their input is the input image is taken from their the radiology department. It is just from their computer, it is transferred to the computer of the radiologist. Yes, the radiologist will open the app, the application of that particular the diagnostic tool. Then just he will drag and drop the image. It will tell where where what is the problem, where is the problem. This tool is developed by us only, but how far? Because it's a big challenge in the field of medicine. We have never achieved, even the whatever I have shown is like looking very quite interesting, but none of them have achieved 100% success. That is a very important underlying concept there. It is not 100%, but we are working towards 100%. So just you can fix yourself in some areas of this research. Just you can think of it how I can help something to improve that, the accuracy. In the normal terms you can say accuracy, right? So there are other properties we can say inside, but generally you can say they increase the accuracy. So maybe interpret broadly incorporate with both detection of the abnormality task and classification task, likely that the abnormality represents a malignancy. Okay, motivation for this one, he computer aided diagnosis for breast cancer, lung cancer, colon cancer. It's like breast cancer is mammogram is an X-ray kind of thing. Just we can take it and find out where is the lump, and whether the lump is just once it is identified, it is whether it is malignant or benign. They have to find out with the tissues, all this. The computer aided diagnostics for lung cancer, thoracic CT scan, and here I have to insist. I have to talk about one thing: HRCT, high resolution CT scan. See, lung cancer or ILD. There is another disease called interstitial lung disease with respect to the lung. But this creates a lot of problems. Even there is no medication for that. Diagnosis itself is a big challenge. You can keep in your mind ILD, interstitial lung disease. Is the HRCT, high resolution CT scan, will produce 300 frames, almost 300 frames of the image. You can say, this vertical and horizontal, the axis it is taking the 300 frames, and the radiologist has to go through all the 300 frames. Otherwise he may miss the probability of the disease ILD that affects normally the tissues. Even though it is similar to a kind of cancer, but it is not cancer at all. But it is similar to them. It loses the function of the lung, and it causes some damage, and there is no proper treatment for that. By diagnosis, what they can do at the final thing, they have to open like open heart surgery. They have to open the chest and take the biopsy of the lung, and they have to send it for the diagnosis. But if we can do that, is actually this is one of the we have tie up with one hospital here in Bangalore, the well-known hospital, and our project is going on. Okay, so this project is very challenging one, like lung cancer, no diagnosis, and in the beginning itself, It's a wonderful thing. Okay, the colon cancer is common thing now. You might have heard about this. Many people are affected by this abnormality of this colon cancer, and just we can see just whatever the images how we can do it. So this organ segmentation, breast immunothoracic images, this organ segmentation, then lesion abnormality segmentation, then feature extraction, then we do classification and evaluation and interpretation. This is our area, right? So just that feature extraction is the segmentation. Of course, it itself is a big challenge. See, just one small in a funny way on my to my research scholars, I say that suppose the doctor trust your research, the doctor trusts your research and say that based on your research he is getting one so he is getting one information, just one patient is having this kind of mass in that particular tissue and he is going for surgery. Your segmentation shows that only small area, but it is above when it is bigger area it covers. So then it is a problem, right? So when he does the surgery, he will remove only small part, but the remaining part is left out. If it is cancerous, it is very dangerous, right? Of course it will not happen like that, but I'm telling that the segmentation, how our algorithm should work, it is not like it is like not a funny thing, just like whatever we give, whatever it comes like that. So we are giving what we are giving for the life of the humans, right? So we have to be very careful. There's four top technologies in medical imaging, artificial intelligence, we know that. Okay, why we need the intelligence? See, if we have this machine, okay, machine, it takes images and just it produces some openness. More than that, when the output is given from the machine, if it gives based on the prior knowledge, that's what we say in artificial intelligence, machine learning. If you go one step above, then internal part of machine learning, you can say deep learning. Then you can say that this using the existing knowledge or the past knowledge, it can combine and it can give the best result to the radiologist. So artificial intelligence, it's a beautiful thing, high quality image supported for diagnosis. And okay, so then radiologists will get very faster information. Patient is also getting right diagnosis because it is not depending on one person's knowledge. I'm sorry, it's not one person's knowledge, it is a collective knowledge, the intelligence, not the knowledge, intelligence. Okay. So augmented reality, there is a two things. No, there are two things. One is augmented, the other one is virtual reality. Virtual reality is not the real one; it is a virtual one. Just we are creating the just virtual reality. I'll tell you. Just you may know that hypnosis. In the cycle, because we have some project with the psychology also. So the hypnosis, they say we are taking into a different world, right? Even though you are not. Very strong, you have to imagine yourself, you are taken into the subconscious mind, like you are a very, that is a virtual reality, then it becomes real, right? So that's what virtual reality, you can imagine, just for your understanding, I'm telling. Augmenting the reality. See, augmented reality is just the reality one only. We are augmenting through sensors. We are augmenting through the images coming from the signals coming from the real-time process, right? We are augmenting it, and we can predict also, right? Suppose, just imagine there is a surgery going on. Right, the doctor is using this augmented reality. With this, he is seeing the thing, and that actually all the input signals are coming, and images are coming of the surgery going on, and the process is happening background and telling that there is a chance for this kind of failure in this particular organ, this kind of bleeding is more in this particular organ, this the vein is getting disconnected, or something kind of alarm can come. The alert will be there, that augmented the reality. We are augmenting the reality to get the best one. That's what we are talking about. That this one, just open site AI system, already we have talked about that. Then the three-dimensional printing that I said, it's mainly for this kind of training the patients. Normally, it's like how the patients have to so more ethical, helping patients understand the kidney tumors and how it can be, just we are going to work on this, all these things to give the study, to give educate the people like that. Cinematic rendering, it is like similar to that, and just giving the best view of that and giving the best. It's also happening. The digital twin technology, see, the digital twin is just like the real one and the digital one, the corresponding digital one. So digital twin is similar to the existing one, but the digital form of the, so the twins, no, we are the real one and the digital one. So digital is the copy of this one, and so they are working on this and it can give best knowledge of this one. So in simulation, so if you talk about, so it's like also it is also like simulation. You may ask, simulation talks about only one dimension, right, or just one part we can simulate and show, but digital twin is the entire part of the simulation, the exact depiction of what's happening in the real object. We are finding it in the digital twin. So if we can imply this digital twin with nanotechnology, nanoparticle, then beautifully we can, with the real tissue and the nanoparticle, then it will be very efficient. But it is not happening so far. Just one of the areas we can develop. So here, so this is also we have worked on that. So medical image with data processing, Hadoop is the best and superb one, but some people may find difficult because it's a process is a little more. It is using, as we know, that it is like MapReduce, Google's MapReduce technology, mapping and reducing, and we can have a number of clusters. And if we implement in cloud platform, really it is very effective, beautiful. So it is from the Apache Hadoop. You can see one little elephant, right? That the person who found his son was playing with that elephant. That's the reason we put that logo as elephant. Okay, so one of the best big data tools. Okay, so Hadoop itself is a big process. Many of us are teaching that Hadoop, also working on Hadoop also. Atlas.ai is one of, this is not completely free. It's a 30-day trial, and we can use it again. So it's a big data analytical tool, mainly for research. We can use export information on each source of data. All right. Okay. Then HPCC is a big data tool. It's why LexisNexis risk solution, and big data tools coverage big data task with far less code. You can just, it's like the graphical ID, integrated development environment is there where we can implement all this. Storm is again from Apache. It is like similar to that, but it is a little more faster. It is, you can see here, even 1,000,000 100-byte messages per second it may handle, in a node may handle. Okay, we have multiple nodes. It's a distributed computing. Okay, there we can see even the batch processing, that Hadoop. When we talk about this Hadoop is batch processing here, and when we talk about this one distributed real-time fault tolerant processing system we have. Okay, so guarantees that each unit will be processed at least once or exactly once. With this, I think I have come is 5:18. Okay, thank you so much. I have given the overall view also, even I haven't taken any particular algorithm to explain. Thanks for listening. So if you have any little questions, whether we can share our knowledge or you can share your experience, some of you are here, you might have come across, so others can listen to your experience. Anything is fine from my side. Thank you.
Thank you so much, sir, for the enlightening presence and the videos, visual learnings. Everything is very nice, and we are having a very clear vision on it, sir. Thank you so much. Dear participants, if you have any doubts, please post the questions in the chat box, or you can unmute your mic and ask the questions. Please interact with our session speaker.
Good evening, sir. Myself, Prashanti. So I'm working on cancer imaging with lung cancer automation.
Okay, ma'am.
So could I make it possible with the X-rays and CTs? So my training feature, like I have now included my feature selection list. Like I'm trying to put out with the chest X-ray as well the CT X-ray. So like the initial biopsy method also will hold the chest part as well. So like all these training data, will that help me to if I pass any one of the input, either the X-ray, chest X-ray or the CT, so will my system able to be predicted, sir?
Yes, ma'am. It's really good with the X-ray alone. You want to have, or you are using both together, ma'am?
I'm training both together, sir.
That's good,
And
ma'am.
I'm also I'm having biopsy data, so I'm also training on that. So like
That you can
like I
take
have
it as a clinical data, ma'am. Then you can this one. What
no,
is
sir,
it?
like the biopsy captured image with this like CT
Oh,
image.
image. Okay, okay. Image you are talking about that. I thought just values, qualitative
No,
one.
like I'm taking everything on an image basis.
Okay, sorry, qualitative. I thought quantitative. Okay, fine. All you
Okay,
are doing
sorry.
with qualitative, it's really well and good, ma'am. And then the HRCT image, sorry, CT images, when you talk about that, if you have HRCT images, ma'am, what I talked about, because we are actually like you only, we are doing one project here with the hospital where we are getting HRCT images where we get more number of frames. So that no
Yes.
data, even from one patient, we get a lot of information. So that would be
Yes,
good.
I do own an data set from the hospital, sir, like from Chennai.
Oh, fine, good, ma'am. That's fine. If you are working with the good and it's really, it will work fine, ma'am. You can reach best result.
Like, sir, is there any other feature that can be like focused more, sir? Like, because now I am trying to augment it for like betterment, but the accuracy level is like still low. So my class for classification is like have set to 14 classes, which is like I have like elaborated my mission to learn more. So like, however can it be? I can bring out the accuracy, sir.
Just you can improve the accuracy you are asking. How many features you have taken, ma'am?
14 classes feature, 14
14 classes. Is there
categories.
any way to improve? Because increase the number of features.
Yes, sir. Like I'm trying on it. So far, I have got
That's
like
great. Ma'am,
300
have you
cases
used
of
deep
data.
learning? Deep learning you are using?
Yes, sir. Deep learning.
Okay, then you can find more number of features, ma'am. Just you can think of in that direction. You can really bring out so many features. Okay, you can bring out
Like
this is like
3D segmented image can also be a feature for my training, sir. Can I take in that way?
just once again, no, 3D image
Uh,
segmentation.
Segmented 3D images. It will show like the extracted nodules
How you got the 3D
already.
images, ma'am? How you got?
Like it is all on the coding basis, sir. Like we have separated three-dimensional segmentation codes, sir.
Okay, I'm not very clear because are you getting 3D image and segmenting or
No, no, sir. It is a
reconstructing?
normal CT. Yeah,
Ah,
reconstruction of it.
oh, reconstructing to 3D, then you are working on that. You are talking about
So
that.
I'm asking, like, I am trying to, like, I have fixed out with this idea. So I don't know, will it work for the
No, no, no,
betterment?
don't say, ma'am. It will work. It will work actually. It will enhance at least 2 to 3% of your accuracy. Don't worry about that. It will enhance, but may not be because just by hearing just one small ideas about that, I can't confidently tell that, say that, but I can say that definitely you can improve the accuracy in all possible ways. This is all promising techniques, ma'am. What we are doing, no, it's all it has happened already. We are all doing. It is working
Yes.
fine. It is increasing the accuracy. So no issues, ma'am. You can do that.
Okay, sir. Thank you, sir.
All the best, ma'am. Good,
Thank you,
good.
sir. I'm candidate from SRM, sir. I'm a student of Chitra, ma'am.
Oh, that's
So,
nice, ma'am. Yeah.
so I captured all this knowledge from ma'am only. So I must thank ma'am for taking this sessions and all on cancer imaging.
Okay. Mhm. That's
Thank
nice.
you, ma'am.
Really, I have
Sir,
to
another
appreciate, ma'am.
question.
Pardon?
Sir, another question from Meenakshi ma'am.
Okay.
How to decide which modality is best suited for our specific research?
Ma'am, just we have to say normally, as ma'am said, if it is a lung, then X-ray and CT is fine. Even there are cases we can't just generally we can't say, but we have to go with MRI also. MRI also we need to go sometimes chest MRI. Even for we say that ovary cancer, they say blood test. Initially, that they go for blood test, then they go for X-ray, then they could find in with the X-ray just we can they can find some mass. That's all. They can find some mass there. Then they will go for CT scan. Yes, of course, that will show the size of the mass in the abdomen. If it is cervical, sorry, I'm sorry, it's ovarian cancer like that. Then we have to go for MRI. And then how in depth of all these things we have to find out. then we have to go for PET scan, ma'am. So it depends on the, especially with cancer, they will not leave with 1 or 2 stages of modality. The modality will take now at present scenario, they will ask us to go for all these modalities now to confirm it. But to work on this, the best thing is if you get data of MRI and to initial in the initial thing, if you have the clinical data, please work on that. Then come to take some features from there. Then you come to the MRI data. Then you work on that. Just whatever the algorithm according to you, you feel even genetic algorithm you can use or any other algorithm, K-means algorithm or nearest neighbor algorithm, whatever you want, just use it, apply it, segment it, and find out the features from there. That you can work on it now. The modality we can't say exactly what. To some extent, we can say depends on the region. And the best
Thank you.
modality for cancer is, ma'am, it's this is PET scan. At present, the current thing is PET.
Thank you so much, sir. Another question from Felicia, madam. Can you please give some insights about multimodal imaging?
Yes, ma'am, multimodality is like, all together we combine. That's what we want to say that, it's like if you come just if you want me to explain in a very simple term, like using one algorithm to segment, it may be helpful. If you combine 2 algorithms, that's what we do normally, right? Currently, we club 2, 3 algorithms and we find the best results. Like that, if you say that multimodalities, like during the if you talk about this nanoparticles, nanotechnology, nano oncology, and when we come to this treatment of that and responding to the treatment, the one cycle itself is using the MRI and PET. So just MRI, based on the result, the patient will go through the this PET scan, and the PET scan will reveal some information. Based on that, immediately we can treat something, give the medicine, nano drug. That's what the algorithm what we talk about, the nano drug, nano particle. Then based on that, immediately we respond. We get some response for that with the PET scan again, or with then next with X-ray or just MRI, whatever it is. Then that's what multimodalities. It is not with one device. One device is giving multiple thing, multiple modalities. We collect the information and just we process the information to give the better results. Because all the time we don't need to use the PET scan, or all the time we don't need to use the MRI scan also, right? That's what we do normally in the computer algorithm processing stages.
Sir,
Thank you so much, sir.
Thank you.
sir, I
Thank you
have
so
one.
much, sir. Yes, yes, ma'am, please.
Sir, I have one doubt, sir.
Hmm.
How nanoparticles works in cancer cells, sir?
Yes, ma'am. See, this nanoparticles, it's injected in the body, correct? So this,
Yes,
it
sir.
goes to the bloodstream. Then it goes to the tissues, organs,
Yes,
tissues in the organs. Okay.
yeah, sir.
So the cell absorbs, our body absorbs. Okay, ma'am. So the nanoparticle has, as I told you, this nanoparticle is like silica coated with some gold particle, whatever it is, then
Yes,
with
sir.
the medicine. So what happens? The radioactive medicine. So it goes to the absorbed by the cancerous cell also. Okay. So then what happens? We just imagine we send the infrared radiation. So only this particles absorbed by this particular tissue, because I'll tell you, the cancer cells have more tendency to absorb more energy. If I talk in a normal layman's term, if you say the cancer patient suddenly he lost weight, you might have heard about it. Suddenly this person lost weight. Why? The cancer cells are eating the energy consumed by the patient, that person. Right. So it takes more, no? So it will take the same kind of thing. It doesn't know. It is going to take the radioactive material. So it will happily eat the nanoparticle. So it has more nanoparticle. It just I'm talking about in a very normal, simple way. So when we send the infrared rays, it's absorbed by this nanoparticle, and it is affecting the nucleus of the cell, cancer cell, because it is absorbed by cancer cells only. So inside the cancer cell, this nanoparticle is sitting, and this nanoparticle is using the taking the radiation and gets burst. The nucleus is burst and it is split. The cancer cell is destroyed.
Okay, sir. So whether we gave the cancer bodies chemotherapy or radiation also, this is also nanoparticles, sir?
No,
Chemo
ma'am.
and
Chemotherapy
radiation.
is a normal drug, ma'am. Normal drug. It is
Okay, sir.
working with all the cells, ma'am. It is well. That's why, if you see the patient who is going through a chemotherapy,
Okay, sir.
before the chemotherapy they will be fresh. After the
Okay.
chemotherapy they will be very tired and dull.
Yeah,
They will
sir.
become more sick because
Yes,
normal
sir.
cells will also be affected. That is not kind of.
Okay, sir.
Radiation also very difficult, ma'am. That's what yesterday even I think doctor talked about that. It's
Yeah,
very
sir.
difficult to locate the particular location, and radiation comes from the outside, and definitely it is a big process. It has to affect only that particular thing and kill the cells. It is very difficult. That's why they want to go for the nano technology.
Okay, sir. Okay, sir. Thank you, sir.
Okay, thank you, ma'am.
Thank you, sir.
Sir, I have a few basic questions actually. Sir, you were
Yes,
mentioning about different technologies which help in the CAD actually, like computer
yes,
aided diagnosis.
yeah.
So you are mentioning about AR and VR. So I understood in a different way actually, like AR and VR can be used in the treatment, like, or for example, if there is a surgeon who is actually trying to do a surgery,
Of
so they
course,
can
correct.
actually imagine and do it. But how does it help in CAD, sir, diagnosis?
Diagnosis also. Oh, okay, you are asking about this. No, no, I am not connecting this with the diagnosis, with the treatment only, because computer diagnosis one part is over. Where in medical imaging, where we are using the augmented reality or virtual reality is helping. What you said is exactly right at the time of surgery. Okay, so it's giving, that's what I said. When surgery is going on, I use the term like that. When the surgery is going on, the using the augmented reality, the doctor will be alarmed. See, this may go wrong, that may go wrong, so it has to change the process, or they have to get ready for other kind of processes for the patient. That's very important. What you said is
Yes.
right.
Yeah, yeah. Okay. Thank you, sir. Sir, my next question is like, so I think many of you are focusing on the medical imaging actually. So most of the problem leads to classification actually. So we need to classifying whether it is a cancerous cell or a non-cancerous cell, like banana management. So apart
Yes.
from this classification, so I asked this question in the yesterday session also, but I think it is relevant to ask to the computer science expert rather than asking to a medical expert actually. So like, is there any other problem domain which we can focus on related to the image processing? I mean, medical image processing.
Image
Like,
processing.
apart from classifications, apart from
Apart from
classification, yeah.
classification, image processing.
Yes.
Okay.
So one problem which from the previous session which I understood, like we can model algorithms which actually help to draw the cancerous part actually, the segment where we can detect the cancer. So it is an algorithm which is going to draw that actually.
Yes,
So we
the
write,
segmentation.
yeah. So that one problem. So apart from this thing, so can we focus on something else?
Normally, the imaging is for the purpose is just for know that whether we have that the patient has some kind of abnormality in that place, right? That is the major issue.
Yes,
So we
yes.
are focusing on that.
Diagnosis part, yes.
What we can do is we can enhance it actually. We can enhance it with the artificial intelligence and using deep learning. Deep learning is best for medical imaging and give the more image. Just the moment itself, the patient is going through the test, the imaging process. He comes out. He comes out not only with one doctor knowledge. He comes out thousands of doctors knowledge, we can say, because we have incorporated the knowledge of thousands of patients already, the existing ones, right? So that kind of thing we can concentrate. Just like artificial intelligence, deep learning we can incorporate. That's all we can say. That imaging part, we want to know what we can do, or even we can predict something from there. See, this kind of image, today it's normal, but it may lead to, because one of my student, the research scholar, is doing like that. So what can be, even for the day he is having one disease, that may lead to other disease, right? That kind of prediction we can conduct. We can find out from the image that one research is doing. He's doing that research. That also we can do.
Okay, thanks. So one last question, like you were
Yes.
mentioning about the segmentation. So I think segmentation is a very important part in the image processing actually. Like so whatever the segmentation algorithm which we use it for the normal images may not be working on the is on the medical images actually. So do you have any
specific
algorithms which can be?
If you ask me, I will say I have got it. So when we have worked with many, then genetic algorithm works well in most of the medical image cases. Genetic algorithm, because but many people have not used much, but still you can try with that. You can see many articles on that, and genetic algorithm works very well, and you can have some predictions from there. That is really good. Just you can try for that.
Yes. This genetic algorithm is on the processing stage. So I'm asking in the pre-processing stage, like we want to do
I'm not very
like a reconstruction. If you wanted to have, because most of the time we get the images on the very poor images actually. So we have to go for a reconstruction of the image. So
okay, I am not very particular about anything now. Just I will see if I will share it with you later in the message through message. I can share it.
No worries, sir. Thank you so much.
Thank you.
So another question from Sudha Ravikumar is: nanotechnology saves the destruction of normal cells to a large extent?
Once again, ma'am,
is nanotechnology saves the destruction of normal cells to a large extent?
No way, ma'am. That question actually, see, nanotechnology is nowhere connected to normal cells. We don't want to deal with normal cells at all. That's why we go for nanotechnology. Nano, in the term itself, I told no, just like 1 inch. If you divide it by 254,000, then what? That one small piece is that nanoparticle. So that nano technology is not for anything related to normal cells. It's only to deal with the cancer cells, where it will be reacting for this radioactive isotopes, like any mechanism, any chemicals. That's all.
Thank you, sir. Next question from Dr. T. Arumugham Maria Devi. Which screening is best for lung cancer diagnosing, either CT or MRI screening?
Ma'am, I can't say like that. When if you say MRI is better, because you will get the 3D parts, and it can give little more clarity on the mass formation in the lung. But actually, for the lung, they can go for in the first stage to confirm. 99% confirmation can come from HRCT itself.
Thank you so much, sir.
Okay, ma'am. Welcome.
Dear participants, if you have any questions, or shall we go to the vote of thanks? Dear participants.
Yes, madam. I am having one question, madam.
Yes, sir. Please. Pankaj Kumar Singh, sir.
Yes, sir. Myself, Dr. Pankaj Kumar Singh from IIT BHU.
Very good.
Your presentation was very nice, sir, regarding so many aspects of the research you have taken. My question is concerned with the nanotoxicity.
Okay, sir. Yeah.
During the cancer treatment, how it will be effect in the treatment of the cancer patient?
So nanotoxicity is a good question, sir. That's what we have. Actually, it's not yet completely established so far. The nanotoxicity is there. That's why even they are having this kind of dilemma whether to go forward or come, and how far it is. It's all under research projects, research process. So we are not able to confirm it so far. And nanotoxicity is there. Nothing is without side effect. Sir. That's what I can say at present current scenario of the study.
Okay, thank you, sir.
Thank you, sir. Thank you for nice questions.
Sir, like one more question to add on, sir. Like now we know like the personalized medicine with artificial intelligence is right blooming. So what could be the objectives of that particular study, sir?
Objective of the particular study?
Personalized medicine.
Yes, ma'am.
With artificial intelligence.
Ma'am, at present level, we can say just the chemotherapy they are giving with the dosage variation. That's all. So the person who has the caliber to tolerate the person's health, other factors they will see. And if he has the caliber to tolerate the particular medicine, the dosage is more. or the patient is weak, the age factor, the older people, and age factor, or some other the comorbidities we say, right? So all these factors will alter the medicine level. So what artificial intelligence, because it is very difficult for the doctors, but what maximum they will do, the milligram, they decide on the milligram. But this will tell this particular patient needs, suppose even the variation may be, suppose 100 mg to 500 mg medicine of the chemotherapy is given. This can tell even this particular patient needs 120 milligram in every week, or the other patient needs 320 milligram because of the other factors and all. Because of the artificial intelligence only we can predict this kind of vast difference, which is not at all possible in the normal analysis stage, normal using the normal analysis process. This is
Sorry,
what
if it
particular
is
patient.
sorry, if it is with the nanomedicine, how could I correlate that same scenario with an artificial
Yes,
intelligence for
ma'am.
nanomedicine?
Nanomedicine, ma'am. Then it's beautiful. We can do it with depends on the level of cancer, the stage of cancer. We can change, no?
So, like
Stages of
only the
cancer.
SEM and TEM material will help me to train on it. So only that data, or like the categorical data should I add on?
Can you please repeat the question, ma'am?
No, so like the SEM and TEM result of like the nano materials, like when I tried out this as an objective, like I met an pharmacist, so he gave me like the data to be handled with artificial intelligence. It is like only on the SEM and TEM results. So I don't find that is like sufficient for my study. So what
Yes.
other categorical data could I add on to strengthen my design, sir?
For this, what is your research area, ma'am?
No, so like it was like for a proposal. Right now I'm working for cancer image only, like lung automation. So like
Okay.
I want to extend it for my proposal. Like I'm trying to put it out with a nanomedicine with artificial
Okay.
intelligence for
Okay.
personalized care.
Ma'am, at present, really, it's good if you don't need to depend more on the data. Of course, we need data. Of course, we need data. We can use whatever the existing data. Whether you can build on this data, whether you can just data, just expo. What I don't know how to say that, because this data only the limited one. The nano is currently work. They are going on. That's working on that process. So don't need to worry about that. Whatever you have with that, you build. Then just you can correlate with the nano. It is better. It's enough, because you don't get more on
On
web
data.
or anywhere. It's under process, so you may not get much.
Okay, thank you, sir.
Thank you, ma'am.
Sir.
Vote of thanks. Yes, ma'am.
Yes, sir. Some of our participants are in need of mail ID, sir. Can you please share your mail ID, sir?
Yes, can I put it in the chat box, ma'am?
Yes, sir. Yes, sir. Thank you very much.
Thank you, ma'am. Yes, ma'am, I have put it there.
Thank you, sir. Dear participants, our speaker's mail ID was shared in the chat box. Please note it down. Ma'am, shall I propose the vote of thanks, ma'am?
Yes, 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 yes.
Good evening to all. I deem it a great honor and privilege to propose the vote of thanks on this occasion. Let me first of all start by giving glory to the Almighty God for making today's occasion a resounding success. On behalf of the organizing committee, I express my heartfelt thanks to Dr. Peter Augustine, Associate Professor, Department of Computer Science, Christ University, Bangalore, who despite his busy schedule has found time to grace this occasion. Thank you so much, sir. The session gives us clear vision on nanotechnology on the cancer diagnosis and therapy. My heartfelt thanks to Professor Dr. P.L. Chitra for her valuable contribution, guidance, and encouragement in all our efforts. Last but not least, I thank all the participants for your cooperation and patient listening for making today's session a grand success. Once again, I thank you all, one and all present here. Thank you so much, sir, for an interactive session and a very interesting visual presentation also. Thank you so much, sir.
Thank you, ma'am. Thank you, everyone. Thank you, Chitra, ma'am. Thank you.
Thank you.
Bye.
Thank you very much for immediately accepting our invitation and having poured the knowledge in this domain for our participants. I think this will definitely help all our research scholars to go into the prospective directions of our this research, sir. Thank you so much for your valuable time. And we request your collaboration in all our activities.
Sure.
Thank you so much.
Sure. Thank you.
Thank you so much, sir.
Thank you.
Thank all the participants and help coordinating the workshop. Special thanks to Egamai today.
Thank you so much,
Bhavani,
ma'am.
Bhavani, Jennifer, and Sasipriya, and all our team members. Thank you so much for your wonderful work. Let us have a effective session tomorrow. Tomorrow we have our session Respiratory Motion Prediction of Lung Tumor Using Artificial Intelligence by Dr. J. Chandra. I hope all the participants will enjoy the session tomorrow also. Thank you so much. Feedback form can be filled and we can wind up the session today.